Hello everybody, and welcome to another video with me, Ro, from Scalpel and Stem. In this video, I'm going to go over some basic terminology for tissue culture. In my last purge, I forgot some people are new and don't know the terminology to tissue culture, and I offered items that weren't for everybody. So we're going to go over all of the different terminology that would be used especially in purges. So, let's get started. The term media can refer to two things. It can either be your planting media, if I'm acclimating, or it can be what sustains the plant life in these jars. My media can be a variety of different colors. Media is usually clear. I add things to mine to differentiate the batches in the pressure cooker, but if I want it to be clear, I can make it clear. Oftentimes, you'll find the rooting media I make is black. This is due to the fact that I add an activated charcoal. If I'm shipping outside the continental 48, my rooting media will be clear. I cannot ship black media to Hawaii, Alaska, Puerto Rico. I like to add activated charcoal. It's not necessary in the rooting process. But as you can see from these variegated fried eggs, these are variegated alocasia fried eggs. They're quite happy in the activated charcoal media. And the fact that it's black makes it really easy to see the roots. Most of my other media will be colored, as it is with this Thai con. This media is yellow. That's usually my Monstera formulation. ZZ. This variegated ZZ is in a different level of hormones than it was for the Monstera delicioso. She's coming along quite nicely. Like every other lab, I do have plants in. Since this is my Monstera delicioso albo, I wanted to make sure that I could see clearly the differentiation in the plant with no adulteration. So, all of my carnivores are in a clear media, and this is because carnivores are so sensitive to any additions in the media. As long as it's in a container with plant growth hormones and media, it's considered in vitro. Subculture means that I will be taking plants out of one media and putting them into another. For example, these Venus flytrap, these are red dragon Venus flytrap, they are not ready to live on their own. They do not have roots, and right now they look gorgeous, but it's just a ball of callus and tiny little babies. And to the untrained eye, this looks dead. What actually happened is I took a ZZ leaf and introduced it to in vitro conditions, and then I grew callus from there. This brown stuff is actually callus. So what you see here, callus, is a mass of undifferentiated cells. You do see a few gray, but the big brown mass that you see that isn't dead is just undifferentiated cells. They're looking for a place to go. I've intentionally kept them in low hormone to keep them this way because I'm not ready to work with them at this moment, and this is a perfect way to do it. Whenever I am ready to work with these ZZ, I'll just add the hormones that I'm thinking of using, and they'll come back out of callus and create actual little plantlets again. So I have my rooted fried egg, and I'm ready for it to come out and be planted. I would do what's called acclimating or hardening. I have made a video on this to explain the process further, but all it means is moving the plant from in vitro conditions to living outside of the in vitro conditions. I hope this helped to clarify the terminology in tissue culture. And as always, I'm here if you have any questions. Well, thanks for watching.